slow-bar cauliflower. So, and recently, sir also released one purple color cauliflower variety. This is the first public sector variety released in purple color. So, and also have the lot of publications in high-rated journals, Cytometry. So, thank you, sir.
Okay, good. So, good morning. Actually, today's lecture is on speed breeding, speed breeding in vegetable crops. When we are talking about the speed breeding, we must accept that the speed breeding in the different crops is not well established, first of all. This is a new concept actually, and whatever work has been done till now, and most of the work are in some of the major cereal or the grain crops, and very few works, 1 or 2 papers recently came in the vegetable crops. But this is a very, very important concept that one you have to understand. First of all, why we need speed breeding? What was the need? Because we have a lot of the tools. We have genomic assisted tool, we have all the transgenic assisted tool, we can modify any trait. But then why we need the speed breeding? If you just see the cycle of the plants for improvement, first of all, what you have to do? You have to do either selfing to generation advancement, or you have to do some kind of the crossing which are having some desirable traits. But when is there any shortcut of the selfing or the crossing or the sexual hybridization? Because you have to develop the progenies. If you do not develop the progenies, it is not possible to undertake any improvement program. Whether it is marker-assisted selection, whether it is genomic-assisted selections, or whether it is based on the transgenes. Because you have to derive your progeny, you have to make some of the mapping population. For transgene, you have to make the progeny, you have to evaluate them. And for any genomic assisted breeding, even you are talking about the haploids and the other thing, you have to have developed the progenies. So first thing is that there is no shortcut to develop the sexual hybridization. We have to go for the sexual hybridization for crop improvement. Now, the things is that particularly in horticultural crops, some of the crops like which are most of the fruits crops are perennial. Although we have the advantage in vegetable crops, in some of the crops we can take 2 to 3 generations per year, like crops like cucurbits, crop like Solanaceae crop. But even the crops like which are temperate in nature, like cauliflower, like carrot, they cannot be grown more than once in a year. But when you are coming to the fruit breeding means breeding of the citrus, breeding of the mango. What is the cycle? Cycle is at least 5 to 10 years for a single generation advancement. But if you want to integrate a particular trait through backcrossing, how much time you will need? You will need 50 to 100 years. By then you will be, now none of us will be here for integration of a particular trait. But if we have some technology which can accelerate the breeding, we can generate instead of 5 years, it can be done within 2 months. That is the concept of the speed breeding. And this concept came from the NASA's efforts, because NASA, they are spending the spaceship to the different international space station and the others. So they are trying to grow the cross species there by modifying different factors. And our scientist has taken the cue from the NASA's experiment, and we have started working on this technology only in the last 10 years only. And majority of the works are mainly done in the Australia, University of the Queensland. They are the major groups who are working on the speed breeding. Then why? Because all of us are aware of the population and all those things, because we have a huge population, world population is growing in a very fast. Suppose a challenge come today, some kind of the, you can say some kind of the breakout of a particular disease came now, today. That disease has to be controlled, and we have to have a product within a very short time. But if we go for the conventional breeding, how much time it will take? Even in the vegetable crops, at least 4 to 5 years. But our challenge is that can we do it within 2 years? Can we do it within 1 year? That is the main thing in the speed breeding. So speed breeding is such a technology that we can improve or we can develop an end product within a very, very short period of time, which we have never thought earlier actually. So suppose in case of the citrus is the citrus greening, Citrus greening is a very, very severe problem. And if citrus greening have occurs, it generally eliminates all the orchards wherever it is happening. And most of the major citrus species, they are susceptible. But resistant source is available, like in case of the Citrus maxima, which is the pomelo. But can we transfer this one? One backcross cycle takes 5 to 10 years. So if we go for the conventional breeding methods, it is at least 50 years. So by that time, it may not be relevant, no? So that is the challenge. Why this is taking so much of the time? Because of the juvenility. Juvenility means this is the phase of the growth before reproductive phase, post germination and before reproduction. That is the phase. It requires certain kind of the photoperiodic requirement, certain kind of the temperature requirement for that, for going to the reproductive phase. So juvenility has to be overcome, and we can do that if we understand the crops and we have certain kind of the facilities with us, because we have to modify certain things here. This is the different ways of the speed breeding, how we can do that one. First of all, modified photoperiod and the temperature. This is the most commonly used technique for the speed breeding, which is being used commercially in different crop plants, and particularly in the different grain crops. Here we are modifying the temperature as well as the photoperiod. Suppose most of the plants, where the plants are the photo insensitive, or where the plants are the long day, no? In that case, we can increase the photoperiod. We can increase the photoperiod to a certain level. And when we are increasing the photoperiod, suppose the natural photoperiod is 12 hours, what we call in natural, in general. But we can increase it up to 24 hours if we want continuous light. So the photoperiod requirement of a certain plant will be met within a half period of the time. Suppose the cycle of the crop is 6 months. But we can do it within 3 months by giving the photoperiod. But this can be done only in case of the long day or the day neutral plant. But similarly, where there is a short day requirement, like amaranthus, no? In amaranthus, that case we can also provide the short day. And here, not only the light, but the quality of the light is also very important, no? What kind of the lights you are giving, what is the wavelength, and all those things has to be standardized. Then secondly, exerting the physiological pressure. You know, naturally, when the certain plants are not getting sufficient water, or they are the water deficient, no? In that case, what is happening? You will see that there is early flowering. So this can be done in some case of the annual plants by giving certain kind of the physiological stress. You are not applying too much of the fertilizer, you are not giving nitrogen, and you are not giving the water sufficiently. Then embryo rescue. Most of the plants have the seed dormancy. So for seed dormancy, we have to wait for at least 2 to 5 months for their germination. But if we can take out the immature embryo and culture them, then the immature embryo can be germinated. And from that germinated embryo, we can produce plant in vitro. And the in vitro plant, we have now techniques to induce flowering in vitro within a very, very short period of time. And after flowering in the in vitro, we can do the selfing as well as the crossing, whatever you need. So instead of the 1 year, embryo rescue can done then that work within 6 months if we have a very suitable protocol for us. Then carbon concentration, you know, increasing the carbon concentration, most of the crops, C3 crops, they are responsive carbon dioxide concentration, and their growth is enhanced and the reproduction phase is advanced. So by increasing the carbon dioxide concentration, but not commonly used, no, till now. Then another is the double haploid. Double haploid, what is double haploid? You are producing first of the haploid plants, then you are diplodizing them. So immediately what you are getting, when you are haploid plants, whatever the composition of the haploids, if you are diplodizing them, what you are getting? You are getting the plants which is 100% homozygous. There is not a single allele which is heterozygous. But it is applicable particularly in case of the cross-pollinated crops, which are highly heterozygous in nature. For those crops, because when we are talking about the end product, no? End product means cultivar or the improved variety. End product, for development of the cultivar or the improved variety, we have to have the complete homozygous lines. Suppose in case of the hybrid development, no? What is the requirement for any hybrids? First of all, one, pollination control mechanisms, and secondly, Inbreds with complete homozygosity. But can you develop an inbred which is having 100% homozygosity through conventional selfing? No. It will take at least 7 years theoretically to achieve around 98 or 99% homozygosity. Even then, 1% heterozygosity is there. What do you mean by 1% in the genomic level? 1% means there are millions of the alleles who will be segregating. So theoretically, practically, it is not possible to develop 100% homozygous lines through conventional breeding. Even after 20 generations of the selfing, there will be thousands of the alleles which will be segregating. But with the double haploid technology, you can do it immediately with 0% segregation. There will be not a single allele which will be heterozygous. So we can develop inbred at the shortest possible time. And in case of the vegetable crop, this is the only technology which has been used till now for speed breeding. And then other biotechnological approaches like flowering gene. Every species, flowering genes are well studied in crop species like Arabidopsis. No? From the Arabidopsis, now we understand what are the homologous flowering genes in different species. So those genes can be modified by using different technology. You can do the CRISPR-Cas for your gene editing so that the flowering time has been reduced drastically. No? So those flowering genes can be done, but again that involves some kind of a genetic modification. No? So those genes can also play a very important role in case of the speed breeding, although not has been done successfully till now. But in future, this technology is going to play a very important role in most of the crops. Huh? Now this is the approaches for just reducing the breeding cycle in different crops. It is actually an example for the tree crops. No? Here we are doing the embryo rescue. You see the plants, we are getting the dormant embryo, we are taking embryo rescue and breaking the dormancy, getting the plants. And in this case, we are providing the suitable conditions. Suitable condition means high photoperiod with optimum temperature. In that case, what will happen? The growth of the plants may be very, very tall. It could be excessive growth will be there. But then again, you can get that one in fruit crop. It is possible to do it grafting somewhere with some rootstock. And you can reduce the generation cycle to it drastically. From 5 years, it could be done within 3 months or the 5 months. It is possible now. No? Similarly, some of the biotechnological approach like I was talking about the flowering genes. I will not go in detail about the flowering genes, just giving you a glimpse. Like by modifying the flowering genes, you can develop some of the rapid cycle population which is having very short period for the flowering. It can be done for the any species. It can be done even for the tree crops. Suppose in mango it is taking 5 years, no? But it could be done within the 5 months also if you can modify with those flowering genes, no? So these kind of the plants can be generated. Then it has also already been studied that even if your rootstock is genetically modified, no? If you are planting, just grafting a scion where you want to do the generation advance, even the rootstock is also transferring the flowering gene protein to your scion. And your scion is getting the very, very early flowering. That is also possible, particularly very applicable in case of the fruit crops. So now here the question will be whether the product, whatever you are getting, whether that is transgene or not. No? That is not well defined till now. We don't know actually. No? Then third one is very interesting. No? Here and again, suppose this is a one tree plant which is having defect for a certain kind of the disease, susceptible to a disease. But we have genetically modifying this one and producing a rapid cycle plant. Then this is the plant where we have the desirable, that resistance source. I was talking about the citrus greening. Suppose here we have the greening resistant. So we can cross this one with this rapid cycle transgene. And if your transgene is dominant, whatever you will get after crossing, that will be transgene which is rapid cycle. So you can do the flowering will come within a very short period of time. So this can be then again crossed with the desirable where you want to transfer the desirable traits. And after certain generation, 1 or 2 generations of the back crossing, what will happen? You can knock out the transgenes. You can just knock out the transgenes, whatever genetically modified flowering genes you have used. You will be taking only the desirable trait, only genes. So your end product will not be a transgene. Although you are using some of the transgene in between to transfer the desirable traits. So this technology is having very promising, although very theoretical till now, not applied. Okay. Now coming to the different techniques of the speed breeding. So most commonly used is the photoperiod and the temperature modified based techniques. And here you need certain kind of the setups. Setups means they are very expensive actually, because you have to control the temperature, you have to control the humidity, you have to control the source of light. Then not only the intensity of the light, your source and the quality of the light is also important, no? So different and most commonly used photo period, generally we are using in different grain crops, that is the 22 hours light and 2 hours of the dark. Because if we are giving 24 hours of the light continuously, they may have some kind of the deleterious effects. Plants also need certain kind of dark also, no? So this is the most commonly used treatment for any speed breeding cycle in different grain crops. And then the initial light calibration means light quality and the intensity. It depends on the crops. Because the germplasms, I am talking about the germplasm. Germplasm means which species you are taking, which species you are studying for the speed breeding. And secondly, not only the species, within the species, what are the genotypes? There could be variation within the genotype to genotype. So you have to standardize the protocol for each and every species. Then when it is standardized in species, it is not sufficient only in species, then you have to go for the genotype specific, no? So it will take lot of time. Earlier what we are thinking, that sequencing has only be done in the rice, it will not be done in the other crops, very difficult, no? Lot of money is involved. Now thousands of the crop has been sequenced, no? Likewise, this kind of the technology will play a very important role in near future, because we will have the protocol for speed breeding in almost every crops, maybe after 10 years, maybe after 20 years, no? But work has already been started. So this is the actual landmark paper in the speed breeding, came in the Nature in the year 1998, no? So this is the Watson, no? Yeah? Yeah, Watson. They have, from the Wisconsin University, they have studied the speed breeding in a set of the crops, 4 crops actually, no? And they have found that in crops like wheat, barley, chickpea, and the canola, they have taken 4 crops. And in the all crops, they have proved that the breeding cycle, this is the natural breeding cycle, 12 hour, 12 hour, no? Here it was around 3 cycles only you can take at the max with the use of the greenhouse. Because even for getting the 3 cycles, you have to have some kind of the control environment. It cannot be done under the natural condition, no? But with this protocol, they have standardized 6 cycles can be easily taken in wheat, barley, and the chickpea. And in case of the canola, instead of the 2 cycle, 4 cycle can easily be taken. So they have reduced the breeding cycles almost 50 to 50%. If it takes 6 months, then you can reduce it to 2 months. So they have not only modified the photo period, they have also taken advantage of the embryo rescue. So they have combined these 2 techniques, no? So embryo rescue means they are taking the immature embryo, drying them, then germinating them, so that easy germination and the early germination. So a single technology cannot be helpful for the speed breeding. We need to combine it with different and the innovative technologies, whatever you have with you, no? So we need to think about that, and we need to design our experiment by ourselves actually, no? What kind of the design can be done. And these are the examples. In crops like brachypodium, barley, and the wheat, you can see the different stages of the crop development. One leaf, this is the stem extension, heading, and the viable sets. And for the heading means reproductive stage. Here in the conventional method, if it is taking 60 days, it is around 30 days in case of the speed breeding. Upper one is the speed breeding, no? So with this speed breeding, here they have modified the reproductive cycle to 50%. This is a very, very significant progress. Instead of the 3 cycle, if we can take the 6 cycles, so within a year we can develop a variety. Within a year, by using the molecular markers, because marker-assisted backcrossing will also be there. So you have certain tools with you, and you are combining these tools so that you are getting more powerful day by day. The breeders are having more things and the more power with them to practice this one. Instead of 2 years, 3 years, now we can develop it. Earlier, what we are talking? We are talking that with the marker-assisted breeding or the backcross breeding, we can develop a variety within 3 years instead of the 6 or 7 years. But now we can say, with the marker-assisted breeding combined with speed breeding, we can develop a variety within 1 year. Okay, this is the beauty of this technology, no? So likewise, in case of the canola, you can see the under normal condition what kind of the growth after a certain period. But within the same period, with the speed breeding and the protocols, you can have this kind of the protocol. But unfortunately, in the vegetable crop, I am not able to give you very successful examples because nothing is available actually. No? Nothing has been done. That is why you have a lot of scope to work on this if you have the facility. But facility will be developed slowly, day by day. No? So we can work in this aspect in different vegetable crops. And I have got only one example in case of the grain amaranth. No? That is a short day crops. Here, the Stellar et al. In 2016, this paper came in the, I think, in Frontiers. No? So they have modified the growing condition to 8 hours light with temperature of the 30 degrees Celsius. So within 4 weeks, they are getting the flower initiation instead of the 3 months. So it has been reduced to a 1/3 of the cycle. No? And what actually their purpose was to develop the crossing techniques. And for development of the crossing techniques, they have to have the continuous flower, no? Continuous flower for an extended period of the time. So they have taken advantage of the long day as well as the short day. So they are keeping some of the plants in the long day so that vegetative growth is continued. And whenever certain flowers are getting over and no flowers are there, they are shifting the plants to the short day condition. So they are getting the continuous flowering for a certain period of time to standardize other techniques what they are doing, the crossing techniques actually they are standardizing. And one example in the pea, no? In the garden pea, here the objective was to develop the real population actually, recombinant inbred line for mapping certain traits. So real population they have developed within the 2 years, 1.5 years actually precisely, no? So they have done certain kind of the protocol using some modified soilless media as well as certain growth regulators, no? And they have done very dense planting. You see 797 plants per square meter, no? Very dense planting, and pea is a self-pollinated crop, no? Even if you get one or two seeds, that is sufficient for you to develop the reel. Because we are practicing the single seed decision, no? Earlier single seed decision. So even if we are getting a single seed, that is also sufficient. And pea is a self-pollinated crop, and that is why there will be the seed set automatically, you know? And the immature embryo you can culture, and they have used some of the tissue culture media. You can see different kind of the media. Then the cycle was reduced to around 1/3, and they have developed the real population within 1.5 years instead of the 4 or the 5 years. No? So this real population can be used for the now mapping of the certain traits. So mapping is helping the speed breeding in certain ways. If you want to do the product development. And speed breeding is also helping the molecular mapping, you know. So these are some kind of the complementary technology. So what, as per your need, you can design your experiment. That is the actually takeaway, no? Now the comparison among the different speed breeding approaches. The most important and the most widely used is the photoperiod. It may not be most efficient. We don't know actually, no? But till now, this is the most widely used. Maybe some other technology, biotechnological based will come, which could be the most efficient, no? Besides exerting the physiological stress, what I was telling you, that we can limit the supply of the inputs, then embryo rescue, carbon concentration, and the double haploids. Then when you are exerting the physiological stress, you are restricting the plant growth area, very dense planting you are doing, restricting the water or the nutrient, then thinning of the plant canopy so that you don't need much of the seeds actually, no? You need only few seeds, 1 to 5 seeds is sufficient, no? So, but it triggers early flowering in the seed set, and it has been done in the rice as well as the pea, the earlier experiment I was talking about in the pea, no? Then embryo rescue, again it involves certain kind of the in vitro techniques. In vitro techniques means under aseptic condition, you have to do the embryo rescue, you have to culture. But here, the important thing that the stage, in which stage you are taking, which nutrient media you are taking, and within the tissue culture laboratory, what kind of the, you can say the environment you are maintaining, you know? What kind of the temperature, what kind of the humidity, not humidity actually, and the what kind of the photoperiod, temperature and the photoperiod. So embryo rescue, which is supplemented by certain kind of the plant growth regulator to enhance the growth, to enhance the flowering, no? And that can also be used, and the some examples are there by the Mobini and the Allset. You can go through these papers, no? And carbon concentration, this, although everybody knows this one, increasing the carbon concentration enhances your photosynthesis and your reproductive stage could be advanced. But this has not been done in so widely because the advantage may not be huge. May not be huge means it could be only a marginal advantage. No, instead of the 5 months, it could be 4 months. No, that is why we have not used this one. But we have other technology which can at least reduce to 50%. Then why we should go for this one? No. Then double haploids. Double haploids is actually important only for the crops which are heterozygous, no? For development of the end product. But for genetic studies, mutation studies, transgene, double haploid is also useful in any crops actually, no? So sometimes we confuse that what is the need of the double haploid maybe in tomato, maybe in chili, because they are the self-pollinated crops. But we have the different objective for the different crops because we need to go for the genetic study for creation of the mapping population. Suppose we are taking the two plants of the chili or the tomato, no? They are self-pollinated, that's okay. But when you are maintaining that one, there may be certain kind of the heterozygosity, no? So whatever mapping population you are developing based on a two conventionally developed pure line, you can say, that will have some difference when you are developing a mapping population, you are taking two DH lines, no? Because DH line, they are the 100% homozygous. The linkage, whatever you will get in the marker, in the segregation, that will be perfect in case of the DH-based mapping population, no? So, but this has been extensively used in case of the vegetable crops, particularly for development of the products, actually, no? Then these are the different application of the speed breeding, that rapid generation of the breeding population. For many things it could be done. Development of the mapping population, mutation study, gene integration through marker-assisted selection. No? Sorry. And these are the scopes. Further, what we can do, that powerful tool to accelerate the crop research and the breeding. You can accelerate it in a very, very high speed. No? Then speeding up the conventional approach, because certain kind of the challenge is now with us that we need to have a certain downy mildew resistance in cucumber. We need a variety, but we have to wait for at least 5 years, 4 years, 5 years to develop end product. But we can do it within 2 years using the speed breeding. Accelerate the crop improvement, high throughput genotyping, then gene editing and the genomic selections. These are the latest technology. They can also be assisted using the speed breeding technology because you have to have the progenies to do any kind of the study, including gene editing and the genomic selections. If you don't have the progeny, you cannot evaluate or you cannot analyze your population, that what kind of the expression they are giving. No? Now, actually, I was working on the DH for a long time, for the last 10 years. So, what I thought that in case of the vegetable crop, when we are talking about the speed breeding, if we do not talk about the double haploid, that may not be a justice, no? Because in the speed breeding, nothing else is available in case of the vegetable crops. So, now coming to the DH, how it can be applied in the crops like cucumber, crops like, you can say, cauliflower, no? Here, when you are going for the conventional methods, you see this is the normal pedigree method, conventional pedigree method. How much time it takes to development of a commercial variety? It takes around 10 to 13 years, because within 6 or the 7 years, we may get the homozygous line, but again we have to go for the multi-location trial for another 2-3 years. But in case of the double haploid, it could be done within 4 years, because multi-location trial takes 2 to 3 years, and for development of the DH line, another 2 years. So within 4 years, you can develop your end product using a double haploid technology, and now we have successful example also in case of the cauliflower from India. No? So secondly, creation of the genetic variability. When we are talking about the DH, it is not only about the homozygosity, it is also about the variability. Even if you don't have certain kind of the variability in your population, you can create it. Because if you take the donor plant as F1, no? If you are taking the donor plant as F1 and the two plants are the different for the different traits, so you can have whatever haploids you will be taking from the F1, each and every haploid will be different. Each and every haploid will be different. So whenever you are just diploidizing them, whatever DH you are getting, each and every DH will be different. So within 2 years, you can create a certain kind of the variability, and that variability you can assess or that you can evaluate, and that could be used as such as a variety. So within 2-3 years or the 4th year, you can develop a variety using the DH-based technology. And based on your objective, you can select your parental lines, what are the traits you need in your variety. No? So this is the different application. I am not going to detail. Then how to develop the double haploids? When we are talking about the double haploids, how to develop it? From where you are going to get the haploids? Haploid cell, what are the source of the haploid cells? Either microspore or megaspore. Microspore or the megaspores, they are the two major sources. Microspore means androgenesis, male gamete, no? And the megaspores means gynogenesis, female gametes. So we can do it in the both stages, either through gynogenesis or the androgenesis. Besides this one, that you can do it from the maternal haploids by using certain technology like irradiated pollen, no? Irradiated pollen, what you are taking here, for pollination, the flower, suppose a plant where you want to induce the haploids, no? In that plant, female flower buds are there. Then you will take for the pollination some of the irradiated pollen of the same species, maybe from different variety, no? Irradiated pollen means what it will do after irradiation? It will make the pollen non-viable. The chromosome will be eliminated. There will be no chromosome actually in the irradiated pollens, no? There will be no genomic composition. But when you are applying the pollens, it will induce the seed setting and the other pollination related physiological mechanism. Means when you are certainly pollinating, so it will induce some of the hormonal metabolism so that the fruit will start developing. Although there is no hybridization, no? There is no fertilization. The pollen tube may grow, it may go up to the ovules also. But there will be no fertilization because the pollen, whatever we are taking, that is the irradiated, there is no chromosome at all. But all the fertilization related activities are already activated in that cell. So some of the seeds like structure you will get after certain time. All the seeds will have some embryos, but whether the embryo will be haploid or diploid, it will be haploid. Why? Because there is no fertilization. Although the fertilization related metabolism or the physiological mechanism has been activated because of the application of the pollen. But here you cannot wait up to the seed maturity. If you wait up to the seed maturity, it will be aborted. Your embryo will be, there will be embryo abortion, no? Embryo may not last because they are the haploids in nature. So in between you have to do some kinds of the embryo rescue, no? After certain stages. Then you know the pollination with some wild species, lot of the example, particularly in case of the wheat and the barley. Even in India, a lot of the work has been done in the Palampur University. If somebody from Himachal Pradesh, I don't know what is his name, I could not remember the scientist. A lot of the work has been done. Development of the haploids using some of the wild relatives, and some examples are there even in vegetable crops. Then gynogenesis. Gynogenesis means you are producing the haploids by germination of the unfertilized ovules. Unfertilized ovules, no? Unfertilized ovule, they will be germinated. Then whatever you will get, they will be the haploids. Then gynogenesis, androgenesis means you are doing developing the embryo from the pollen, you can say in the simplest form, that is the pollen grains, no? But when they are mature pollen, they may not germinate. We have to take in some of the earlier stages of the pollen development, maybe in the microspore genetic. And recently, the centromere mediated, that CNH3 technology came for development of the double haploids, which is also been practiced in different crop, including Brassica oleracea. So when irradiated pollen, I think this is clear for you. I no need to much explain. Wide crossing and the irradiated pollen, basically we are developing the maternal haploids, no? Whatever haploids we are getting, we are getting from the maternal cells. We are not getting from the paternal cells, no? Then these are some of the examples in the vegetable crops using the wide hybrids like Solanum nigrum when it is crossed with the Solanum luteum, because they are not crossable with each other. But the pollen of the Solanum luteum, it is inducing the process of the fertilization, and you will get some of the haploids from the here Solanum nigrum. Likewise, Solanum tuberation when it is crossed with Solanum fureza, and CPRI, some of the works has been done, and they have developed some of the Solanum tuberation haploids by crossing it with Solanum fureza. These are the two successful examples in case of the vegetable crops by using the wide hybrid for development of the double haploids. No? And this is the gynogenesis. This is actually a picture from our laboratory only. Here in cucumber, we have done some of the gynogenesis work, means some of the unfertilized ovules. I was talking about the unfertilized ovules. But can you isolate the unfertilized ovule as such? It is not possible, no? So what we are taking? We are taking the unfertilized ovary before the anthesis, no? Ovary means the unopened flower bud. It is very easy in case of the cucumber because male and the female flowers are inseparate. They are not hermaphrodite, no? So we are taking the female flower buds and we are culturing them. And whatever see, the germination are coming, they are coming from some of the ovules. And after the germination, you will get some of the embryo-like structure from those ovules, and they should be haploids, but we need to check the haploid level because we are not sure, because we have some of the diploid cell around also, no? But in most probably, in most 90% of the cases, they will be haploid, because generally this kind of the germination do not come from the vegetative cells or the somatic cells, no? And this is the development of the haploid through gynogenesis, and several plants have been developed in different laboratories, no? And this is the limitation of the gynogenesis, because when you are going for the gynogenesis means you are taking the unfertilized ovary, no? So in certain cases, in the number of the ovules per ovary is very limited, many species, no? But in crops like cucumber, it is abundant, no? But it may not be abundant in cauliflower, it may not be abundant in carrot, no? So we have to need the number of the whatever ovary and the ovule are the very less, that is a limitation. But when you are going for the androgenesis of the microspore culture, you are taking the anthers and you are taking the pollen grains. Even within a single anther, there are millions of the pollen grains of the microspore are there. So within a single plate, if you culture, you can get hundreds of the haploid plants, but that cannot be done with the gynogenesis. You have to do a lot number of the cultures, and the number of the ovary, you have to take a large number, and it will take more time to develop a large population. But the thing is that the androgenesis and the gynogenesis, they are the genotype specific. Suppose in case of the gynogenesis, what are the successful example of the gynogenesis? Onion, no? Onion, that cannot be done through androgenesis, no? And successful example of the androgenesis, brassicas, no? Brassicas, but brassicas are not suitable for the gynogenesis. So we need to know which technology would be suitable for one. It is not necessary that androgenesis could be done in the onions, no? Because no report is there till now. Although we are trying to do something, but we don't know whether we will be successful or not. We may be successful, we may not be successful, no? So these are the crop specific. What will be respond through androgenesis, what will respond through the gynogenesis, no? And this is the male gametes you are taking through the androgenesis. But what is happening in the androgenesis, if you just leave the pollen cells in the normal flowers, what will happen? The microspores, they will, through meiosis, they will develop into the pollen grains. So what you are doing in between, so that they are not developing the pollen grains, but they are developing into the embryos. Some kind of the symmetric division is happening in between. How it is done? Because in between you have to do something, no? Naturally it could not be germinated, no? So you have to apply certain kind of the shock treatment, which could be in terms of the temperature, which could be in terms of the some of the growth regulators, some of the growth retardant, which could be in terms of the giving some kind of the other electrical shocks and all those things. So that the normal your gametogenesis pathway is diverted to the sporogenesis, and normal, then there will be symmetric division and you will getting the haploid embryos. And this conversion from the switch off from the gametogenesis to the gametogenesis to the sporogenesis is very, very critical. And this is the cross specific as well as the genotype specific, and you need to standardize this stage, you know. If you can standardize this stage, that how that switch off happens, then you are successful in developing the haploids, you know. It is well established in case of the brassicas. Brassicas, even anther culture has been done in crops like solanaceous crops, in the chili and the potato, and we have a lot of the results using the anther culture, and the haploid development is a very common phenomenon in these crops. But when you are going for the anther culture, there are two techniques of the androgenesis. One is the anther culture, another is the microspore culture. Anther culture, you are going for the only anthers as such, no? When you are taking the anthers, along with the microspores, there are some of the diploid tissues are there. So there will be the chances that whatever germination you are getting, they may not be haploids actually, no? They may be some diploid, they could be triploid, they could be some other things, no? But in case when you are doing the microspore culture, means you are taking certain kind of the filters and you are isolating the microspores, and whatever you are culturing, only the haploid cell, nothing else is there in case of the microspore culture, because they are the liquid based culture. I will show you some of the photographs, and whatever germination you will get, they will be haploids. But even if they are the diploid, they will be the auto-diploidization. Auto-diploidization means haploid cell will be diploidized automatically to give those plants, and those plants actually desirable, because one step is not required, because after development of the haploid, you have to diploidize them by application of the colchicine or certain other treatments. But if you are getting the diploid cell from the microspore culture, that is advantageous actually, no? And this is the normal process of the microspore genesis and the gametogenesis, how it is happening. I think it is not within the scope of this course. And this is the isolated microspore culture. And this is the kind of the germination what we are getting. These are some of the photographs from our laboratory only, no? This kind of the haploid embryo, you see some of the microspore they are growing, but all the microspore, although they may be growing to certain points, they may not germinate and give the embryo, no? So only after in a plate you may get 20, you may get 30, you may get 100 also, no? So based on your protocol and based on what are the treatments you are getting and what are the genotypes you are using, that is also very important, no? And these are the response of the cauliflower, different cauliflower genotype, why I have given, because the genotype, some of the genotype you see, there are 100 of the microspore, no? But in some of the genotype, there are only few, 2, 3, 4. So this is very, very genotype specific, and you need to know which genotype is responding. If you want to work on the microspore based culture, you have to identify some of the genotype which are highly responsive. Only those genotype can be used for other genetic studies, for the further genetic studies, you know. And the developmental stage of the microspore is very important when you are going for the microspore culture, no. Generally it is called the late uninucleate to early binucleate stage, because there will be, otherwise there will be tetrad formation after some times, no. So you have to culture the microspore in this stage to get the haploid cells. How you can do this one? Through fluorescence microscopy. But can you do the fluorescence microscopy every day? When you are working in a lab, is it practically possible to take your birds from the field, isolate the microspore and see it under the microscope? It takes a lot of time. So we generally, what is the time? I think I am on time now, no? Still I have time, no? Okay. So you have to, what you have to do, you can correlate the bird size with the developmental stage of the microspores. In a particular genotype, you see some certain kind of the birds, you will take 2, 3 kind of the birds in the earlier stages, no? And you correlate that this bird size is giving maximum number of the microspore which are having the late uninucleate or the early binucleate. So that bird size you can use later on, because the number of the microspore is not a issue here, no? Because there will be the millions of the microspores, no? So you can take some variable kind of the bird size also, so that some of the birds will have the congenial microspores, no? If you don't have the facility for the fluorescence microscopy. And these are the different kind of the pre-treatment. Generally the temperature, pre-treatment, this is the most important and the most widely used, particularly in case of the brassicas, and a cold treatment of 4 degree followed by shock treatment of around 32.5 degree, no? That is the most widely used in case of the all brassicas, including the Brassica oleracea, Brassica nepus, and the oilseed brassica. And this temperature pre-treatment is actually triggering the development of the microspores mediated embryo development, no? They are diverting the phase of the gametogenesis to the sporogenesis, this temperature treatment. Beside temperature, sucrose, high level of the sucrose can be used, no? Nitrogen starvation, osmotic stress, and all those things can be done. But most of the things are generally theoretical. Only the temperature pretreatment that is most commonly used, because that is practically very convenient for us, no? We cannot do the X-ray and all those things every time, no? So this is the temperature treatment which is used for the microsporogenesis in the cauliflowers. And actually I am working on the haploids for the last, you can say 7 to 8 years, no? So we have done a lot of the product, we have a lot of the things here, particularly in the cabbage and the cauliflowers, no? And we have standardized different media for the microspore, because after getting the germination also, how you regenerate the plant, how you mass multiply the plants, how to develop the shoots, each and every step has to be optimized in your own lab condition. You cannot take the protocol from the certain labs, it may not work in your lab. Those who are working on the tissue culture, they know these things, no? So you have to do it in your own lab, and it takes some time, some patience actually. When you are working on the tissue culture, it tests your patience actually, no? How much patient you are, it will be judged by if you are successful tissue culture worker, no? But I am not undermining the genomics people, no? They are also have a lot of patience. So these are the cycles. This is the CNH3. This is not much relevant for you. I think somebody else maybe taught you in the genomics related classes in the transgenics, no? The CNH3. Here you have to modify certain histone protein, so that whatever pollination you will get, after pollination from the mother cell, you will, whatever seed will be produced, but all the seeds will be haploid only. Okay? This is also very beautiful technology, but it involves genetically modified technology. And again, we are not clear whether the CNH3 derivatives are the, generally we are considering it now transgene in India. But scientists are arguing they are not transgene actually, because there is no derivative of the modified gene here. Then after development of the haploid, main challenge how to detect whether it is haploid, whether it is diploid. For that you have to have certain kind of the technology. And some conventional methods like vegetative and the floral parts, by seeing the vigor, the haploids will have low vigor and the diploid will have some kind of the higher vigor. But these are very approximate. You never know whether it is accurate or not. Stomatal count, stomata size, because suppose in a controlled diploid species, you see what is the size of the stomata. If some of the plants are the haploid, they will have some kind of the reduced stomatal size. They have the small stomata. But the tetraploid, that could have larger. Then again, that is approximation, you never know. Because that could be the tetraploid, that could be triploid, that could be the pentaploid also. Because when you are working on a very rich nutrient media, there will be certain kind of the diploidization automatically. Because when you are going for the liquid culture and the microsporogenesis, we are using the aniline media with 13% sugar. Normal tissue culture media, it takes how much sugar? 4.5%, but here we are taking very high sucrose concentration. And besides, a lot of the hormones are applied. So there will be certain kind of the, you can say, auto-diploidization. There will be some of the aneuploids also in between. So you have to identify. So these methods may not be very effective for accurately identify. Then you have to do the flow cytometry analysis for accurate identification. What is the flow cytometry analysis? That actually it analyzes the quantity of the DNA in certain developmental stages. Quantity of the DNA. If the haploid have the DNA of this much, if you are having, you will get the HPLC kind of the peaks. So suppose this is the haploid. This is having two peaks here. One is here, another here. So the diploid, it will have the peaks. The first peak will be just double of this position, and the second peak will be double of this smaller peak. So based on that one, you can identify accurately whether it is haploid, whether it is diploid, whether it is triploid. Even the aneuploid can be identified using the flow cytometer. Then flow cytometer is a machine. It is a very expensive machine. We don't have any flow cytometer in IARI, so we have to do it from some CSIR or the DU, that laboratory. And this is the characterization. These are. And when you are going for the microspore culture, why I am showing it? Because you will get different kind of the ploidy level here. It is not that everything will be haploid or everything will be diploid. There will be haploid, certain will be diploids, they may be triploid, pentaploid, polyploid, or the mixoploids. They will have the different kind of the polyploid. Even aneuploid and the chimera you will get. So you have to precisely identify what are the haploids and what are the diploids. And this is the flow cytometer. This is the machine actually what we are working earlier, and we are working on the CSIR IHBT, one CSIR institute. Through some agreement, we are doing the samples there. And then diploidization of the plant, another challenge. How after developing the haploids you are going to diploidize them? For that, different chemical agents, all that microtubule, that formation, they are enhanced like colchicine, origylene. Among all those, colchicine and the origylene, they are the most commonly used for development of for diploidization of the certain kind of the cell. And these are the certain kind of our protocol for development of the haploid and the double haploid in case of the cabbage, as well as in case of the cauliflower. And you can see these are we have all the DH population in the cabbage as well as the cauliflower. And you see kind of the uniformity the plants have. In the normal open pollinated plants, they will never have this kind of the uniformity. Even one leaf is this orientation, all the plants have leaf in that orientation. This is the beauty of double haploid. And this is the DH2 population. After selfing, we are maintaining this in the, basically these works are in Katrai only, IARI region. I was earlier there in the IARI regional station Katrai. And the most important part is that using the double haploid, we developed the product also. And recently we have developed a hybrid in Pusa Snowball hybrid too. This is the first double haploid based hybrid in India, you can say in any crops, any vegetable crops. So we are successful in developing. The story is now completed, from development of the protocol, from development of the haploids. Now we have developed the end product. But if we wanted to develop this kind of the product within the conventional method, it takes a lot of time. We have developed the inbreds within 2 to 3 years, and we have evaluated the inbred. whatever inbred they are giving the good horticultural traits, we have used that as a male parent for crossing with other CMS lines. So this is the first CMS based as well as the double haploid based upon hybrid in vegetable crops, and this is now been notified for the its commercial identification. No? And these are the certain limitation for the speed breeding. First limitation is the development and the availability of the protocol. We don't have protocol for all the species. So if you are working in some crops which don't have any protocol, then you have to standardize your own protocol, no? Then the setup it required, they are very expensive, no? Kind of the light requirement, day length, and all those humidity, temperature, you have to do that on. But when you are stressing the plant, you are forcing the plants, no? So there may be some kind of the disadvantage. There will be some kind of the adverse effect on the plants also, because naturally it is not happening. You are forcing them. Suppose if somebody, you are writing a thesis within 6 months. If your chairman told do it within 2 months, so what you will do? You will not sleep in the night. You will not take food. No, you will starve. No, your health will be affected. No, so plants are also having the same mechanism. No. So if you do that one, so they will have certain kind of the physiological stress, but we have to analyze all those things, what is happening within them. But our main purpose is the progeny generation. We don't need 100 or the thousands of the seeds. We need only 1 or 2 seeds, maybe 3 seeds, no? So that is okay for the plants, no? I think that's all. Thank you. If you have any query, you can tell. Yeah. Yeah? Can? That study has not been done. No, nobody has done that kind of study. We have to standardize that. उसमें कोई किया नहीं अभी तक. Until and unless you do some experiment, we cannot say that this could be done. No, but again you can do their physiology, and based on their physiology, you can design it.